Magical.
Talk science to each other across the country and around the world. What we did in putting this program together was try to identify folks who are at relatively early stages in their careers. Speakers who have enough experience in particular areas that they can inform and enlighten a broad community and speak to experts as well as non-experts, and give folks an opportunity as they're establishing themselves to connect with other C-DEBI researchers and think about new problems and get insights again across the community towards work that's currently underway. Beth Orcutt is a perfect example of the kind of person that we had in mind when we thought about putting the speaker program together. I've had the pleasure of sailing with Beth a number of times over the last few years, and she's interested in a lot of different kinds of problems and is keenly interested both in the science side, in the technology, and then also in communicating the excitement and the implications of her work to diverse audiences. Beth did her undergraduate degree. I hadn't known this before, but she majored in interdisciplinary studies at the University of Georgia with an emphasis on marine geochemistry. She stayed on for a PhD and finished that in 2007. She did a postdoc at USC working with Katrina Edwards, and then has been in Denmark for the last year and a half, working at Aarhus University. So now she's come back to the U.S. She's starting a researcher position at Bigelow Laboratory, which is very exciting. I guess officially she starts in April. So we've caught her while she's in transition, coming back to the U.S. I also want to note that Beth has really been instrumental in her outreach activities in connecting C-DEBI and marine microbiological research in general to people across a range of ages and interests. She is the person responsible for much or all of what you see at the microbesareawesome.com website, and she was virtually fully responsible for really developing the Adopt a Microbe program, and with help from a lot of people, but most of the work she did herself. She's maintained that program and expanded it and run it now on multiple expeditions. And I think it's a great example of how scientists can connect their research to the world at large. So without any further delay, let's welcome Beth Orcutt as our next network speaker.
Am I good to talk here?
Yes.
Okay. And can I move this video screen out of my way so I can see it? Well, I want to thank all of you in the room and all of you watching on the web for taking some time out to be here and listen to my talk about life in the subsurface, and particularly near that interface of the sediments and the oceanic crust. And I will talk to you about that today. How do I get rid of that? One second. Great. Okay, so hopefully we can all follow along here, and I will try to do my best. There's a little bit of some font issues, but I hope we can proceed through that. So as Andy said, I am currently employed as a postdoc at the Center for Geomicrobiology in Denmark, working with Bo Jorgensen and a number of other scientists on life in the deep biosphere. And I also previously worked here at USC, and a lot of the funding for the work that I'll be talking about today comes from the National Science Foundation and also some from private foundations like the Moore Foundation. So I want to give them some credit at the beginning for helping us do this kind of science. The picture that you see here is of the Jason ROV picking up a sample of seafloor basalt from a seamount, I don't know, maybe about 1,500 meters below the seafloor. And I really like this picture because it kind of captures some of the things that I like to think about, of these kind of biofilms growing on rocks and the interaction of life with the oceanic crust and within sediments. And so I will talk to you about a little bit about that today. Biosphere, let's start there. This is a cartoon of how I sometimes like to think of what the deep biosphere looks like and the different types of habitats that one might consider at the bottom of the ocean. So, let's see, close to land here, you would have relatively shallow sediments of the shelf, continental shelf, etc. And you can have very interesting features there, like methane seeps releasing methane into the water column. And then as you get further away from land, out towards the abyssal plain, You get into areas where there's much shallower sediment on top of oceanic crust that is forming at mid-ocean ridges and hotspot volcanoes. And so I'm kind of interested in the connection between these different types of habitats, between water and sediments, between water and the rocks, and between the rocks and the sediment, and how fluids are moving through oceanic crust and changing during fluid rock reactions. And I put this phrase here, an unseen majority, because these type of environments way out in the ocean are actually an area of a lot of life, and I'll talk about that in just a moment. Another way I like to think about the ocean and the habitats where we're finding life in the marine environment, you can also think about kind of a breakdown of these habitats based on light and how big these different types of habitats are. So for instance, the photic zone of the water column only extends a few hundred meters or less into the water. And so once you get below that depth, there is no light. And so the source of energy in these types of systems is relatively different. It can be based more on chemical energy than on photosynthetic energy. And the actual volume of this part of the ocean where sunlight doesn't penetrate is actually much larger than the photic zone. And then as you move deeper down into the underlying sediments and even the ocean crust, you have these massive reservoirs where life could potentially exist. So I want to know what's down there and what it's doing. Now there was kind of this paper that we often turn to that came out in the late 90s that did some relatively simple back of the envelope calculations to try and quantify how abundant is life in these deep environments. And so this kind of information was based on collecting samples and then looking at them under a microscope and counting how many of these little fluorescent dots, which would be the microbes, there are, and trying to actually quantify how abundant these different types of cells were in these samples. And this is not a trivial task. Collecting this data that was generated from sediments from all over the world, but mainly close to the continents, this kind of calculation came together that the number of microbes on Earth in total, if you break that down, a large fraction of them is actually these microbes are found either in the oceanic subsurface, here 55%, or in the terrestrial subsurface. I put a little asterisk here because I want to point out that these calculations were only based on marine sediments and didn't include oceanic crust in the calculations. If you remember back from the slide before, there's actually more rock down at the bottom of the ocean than there's sediment. And when you scale these numbers up of how much carbon would be trapped in these microbes as living biomass, it could be a very significant fraction of all biomass carbon on Earth. So we really want to look at this more closely. Now, I don't think we should exclude rock from these calculations because the rocks are actually very active at the bottom of the seafloor. I borrowed this figure from Andy Fisher, nice cartoon illustrating how fluids, or how we conceptually think about how fluids move through the oceanic crust. And this is a system where you can have exposed outcrops of oceanic crust that are blanketed by sediments. And these sediments kind of act as a blanket, preventing fluid movement out of the oceanic crust. And it's constrained and then comes out or goes in at these outcrop features. And as the fluid moves along, it gains heat, it changes chemically due to fluid rock reactions. And this could be an interesting place for life to exist. You probably know about hydrothermal circulation in fluids in the oceanic crust from looking at science documentaries on TV of hydrothermal vents, all this hot water coming spewing out of the seafloor. Well, there has to be a lot of water going into the seafloor to support that fluid movement. But we haven't really studied this recharge zone very well. And I think we should because when you think about oceanic crust, it could be it is the largest aquifer on Earth for water movement. And the amount of water moving through the oceanic crust can circulate the entire volume of the ocean on the order of a couple 100,000 years. You can't see that number very well because of the font problems, but so geologically speaking, this is relatively important in terms of moving heat and chemicals from the lithosphere into the rest of the Earth system. So how do we study life in rocks? How would one do this? So this is another picture from a submersible doing work at a seamount where you have oceanic crust exposed at the seafloor. And you can do a variety of things like collect these rocks or put out deployment experiments. And if you'd like to know more about that, here's some references you could look at. And what we're trying to do is see how these rocks weather and try to identify the microbes that might be involved in that. So you put down an experiment, you collect it a couple years later, you could look at that, and you can look at it up close and see different features within these rocks. So for instance, this is a basalt going from a glassy to a more ground mass situation. And you would take these and do a variety of different tests, like putting staining or applying a DNA stain and trying to count how many cells are living on the surface of this material. So this is kind of the process that we might go through to study life in these kind of systems. So unlike, let's say, a rainforest ecologist who can go into the forest and walk around and count all the different trees and different types of trees and come up with an idea of what is in this environment, we as marine microbiologists are a little bit more constrained on how we do this. And because the organisms we want to study are so tiny, and pretty much all microbes look the same, more or less, so you can't really identify what a microbe is just by what it looks like. So we rely on looking at the genetic information that we can extract from cells in the environment. And then we use that genetic information to infer what these organisms are based on how they're related to other things we know that are in culture. So for instance, this is the tree of life that was put together back in the late 90s, showing the genetic relation between the microscopic bacteria, microscopic archaea, and then eukarya, which is all the other life, like we are here in the little branch for Homo. And so what we want to do is go out in the environment, collect samples, and then extract DNA from these samples and compare that DNA to try and identify who's there and how abundant different organisms are. And we want to look at several different types of habitats to try and see how they might be similar or related to one another. So sorry again for the font here. Several potential habitats we might look at. This is a cartoon from a review that Katrina had put together a couple years back now, pointing out different features that one might be interested in looking at. So you have the mid-ocean ridge, and then as the ridge ages and moves away and gets buried, you have areas where you have sediment cover, outcrops where water can go into the crust or out of the crust. And so we want to look at as many different types of environments as we can to see how they're connected and how abundant life is in these systems. Well, how do we get into those deeper samples? We can't just go there with a submarine. We need to do something more drastic, like use the ocean drilling vessel. So this is a picture of the JOIDES resolution and a picture of a drill bit, which would be about this big around. And we use this to collect very long cores of deep sediments and deep rocks, which we can then investigate with our techniques. One problem that we face as microbiologists, especially interested in oceanic crust, is that oftentimes during the drilling process, the samples that we really want, where we think fluid is moving, we may not get back. We can see that by, for instance, looking at the size of the hole that remains after we drill. So ideally, the size of the hole, this is in inches here, would be about roughly the same size as the drill bit. But you can see in some areas there are parts of the hole, this is in hundreds of meters probably over here, sorry that the scale got cut off. You can see there's areas where there's big washout of the hole. And these are probably areas where there's lots of fractures in the rocks, probably where lots of fluid is moving, and we've lost most of that material. It's just disintegrated while we're drilling. In addition, this is not really a very sterile process, drilling. And so we have to come up with some pretty severe ways to try and remove contamination from the outside of our samples to get at what's truly inside of them. And so we also want to come up with other methods to really get at what's going on in these deep sediment and deep crust. So one way we've done that is through the use of subsurface borehole observatories, which we are also referred to as corks. This is a nice cartoon that was put together recently for a Science News article, kind of showing you the concept here that we would go back and instrument a hole that we've drilled and leave it there for a long-term observatory. And these instruments can include temperature sensors, pressure sensors, microbiological experiments, chemical samplers, etc. Really, the sky's the limit in terms of what you want to put down there as long as it'll fit. And then we can go back out there every year or so and change out our experiments and download data. And so one way we might do this is a figure of a observatory project happening on the Juan de Fuca Ridge Flank off the coast of Seattle. And this is kind of a cross-sectional representation of what we might do. So imagine this is the sediment surface, and this is what would be the underlying buried ridge. So you've got about 200 meters of sediment covering about 3.5 million year old ocean crust. And we have a series of observatory holes along this ridge flank, and we think fluid moves kind of in this direction. So we can use these types of observatories to study how does the microbial community in the oceanic crust here change as fluid moves along the system. And going a little bit deeper into how we do this for microbiology experiments, inside of these observatories, we can deploy what we call microbial colonization experiments, which are basically different types of minerals that would replicate the oceanic crust in this environment. And we then pump the fluids in that basement environment through these chambers. And then we can go back and collect these years later and see what kind of microbes colonized the different materials that we put down in these holes. So to talk a little bit more about this Juan de Fuca environment, this is a picture showing you kind of where the system is. You have the Juan de Fuca ridge flank here, and our study site is here on the eastern part of this flank. Zooming in on this area, and again, kind of looking at a cross-section, you can see this buried ridge system. And this is a system that's been studied for several decades. And I'm going to talk to you a little bit about some experiments that were deployed for a period of 4 years to show you what we can do with these cork systems and how this teaches us something about microbiology. So what I'm showing you here is a record of temperature and some various chemical constituents that were measured within a borehole on the Juan de Fuca system. And when this borehole observatory was initially deployed, it didn't actually seal properly. And so you had a condition where seawater could go down into the borehole, and we can see that based on the temperature, which was quite cool and very sulfate rich. And so this was a condition for the first couple years of the experiment. And then eventually, the conditions within the bottom of the borehole turned around enough, the fluids gained enough heat from the surrounding basement that then they could blow out of the hole. They became overpressured and the water wanted to come back out. And so we can see that it returned to more normal environmental conditions of being warm, about 65 degrees, reduced sulfate, higher metals like manganese and iron. So even though the experiment didn't work as planned, it actually was a kind of a serendipitous experiment that allowed us to study the transition from a cold oxic crustal condition to a warmer condition. And this is something that would happen naturally on an ocean crust system over, many years, and we kind of captured it within a couple years. So we could take the colonization experiments that we had put in that hole for 4 years and break off little pieces of these different surfaces. For instance, here you have basalt, denite, pyrite, and biotite, and look at these under the microscope. And what we can see is that there's definitely cells that have colonized our experiments. So the deep basement is not devoid of life. There's things that will grow if you put down things for them to eat. And we can also see these very characteristic twisted stalk features. We know certain organisms make these twisted stalks under conditions of a little bit of oxygen, cold temperatures, and some iron around to support iron oxidizing bacteria. So we see some evidence that we had iron oxidizing bacteria down there. But when we take these materials and we extract DNA from them, the breakdown of the different bacteria that we find is quite different than what you would expect from a rock, for instance, in oxygenated seawater, where you would find these iron oxidizing bacteria. Instead, what we find is a very high abundance of another group of bacteria called the firmicutes. And it's unclear what these organisms are doing. They're not phylogenetically very related to known organisms, so we don't know, are they nitrogen cyclers, are they sulfur cyclers? This is something that we're trying to understand more now. But we don't find any evidence for live iron oxidizing bacteria. We don't get any DNA related to those organisms. And so perhaps these twisted stalks were a fossil remnant of iron oxidizing bacteria that lived in the borehole when it was cold and oxic. And then when the borehole transitioned to a warmer condition, those iron oxidizing bacteria died out, but they left their twisted stalks behind. One other intriguing thing for me is that some of these firmicute type bacteria are very closely related to sequences that have been recovered from terrestrial mines, like gold mines in South Africa, and also from spore-forming organisms that are thought to be delivered from hydrothermal vents, and they are in spore form. They get buried in sediments, and if those sediments get reheated, they will grow back again. Excuse me 1 second. So, There's a slide missing. So this is kind of an overview of what we could do with those corks to understand what's happening within the basement condition. But as I mentioned at the beginning, I'm also interested in what's happening in the overlying sediments, and if there's a connection between this basement environment and the sediment environment. And some early indication that we could point to suggest that there may be some connection between these environments comes from an early work done in the same system, excuse me, where as you look towards as you go through deep sediments, down to about 260 meters to the basement contact, you see both an increase in sulfate thought to be coming from basement, and you can see a stimulation of both the abundance of bacteria and archaea and also of activity. So for instance, they were measuring sulfate reduction rates, and you can see this little uptick in activity as you approach basement. This study was done in an area where fluid is thought to be coming out of the oceanic crust, and it's already probably very warm. One of the things we're also interested in is how do microbial communities living at the sediment-basement interface potentially change as you transition from the situation where water is going into the seafloor and moving along and then coming out. And so during IRDP Expedition 327 in 2010, we did a transect of coring from what's called the Grizzly Bear outcrop. And this is in that same Juan de Fuca system. We think water kind of goes into the seafloor at this outcrop, moves in this general direction, and comes out of the seafloor around here. And so we did a series of coring right along the edge of this outcrop and really try to collect samples from the sediment basement interface here to see if there might be a change or stimulation of the microbial community in the system. So a cross-section here through that same seamount. This is the seamount itself and the sediment, and you can see the location of our coring sites. And I try to use the same color scheme so you can follow along. So our expectation is that as we move further northeast along this outcrop, that we would start to see warmer temperatures and a transition from basement conditions that are relatively oxic and cool to maybe something warmer. Well, it turns out that if you look, for instance, at the chemistry of these holes, and already very close to the edge of this outcrop, we see already that the crust is already quite reactive. So we can see this, for instance, by looking at calcium. Calcium would be coming out of the basement into the sediment. And so if basement wasn't reacting quite vigorously, then this profile would be more straight up and down. But you see that it increases towards the basement interface. And so this indicates to us that there's already quite significant fluid rock interaction happening, even very, very close to where we think water is going to the seamount. So what that tells us is that actually our idea of how water moves through the system is way too simple, and it's probably much more complicated. You probably have water moving around in many different directions and not just going to the northeast. So, for instance, we couldn't see any nitrate in these samples either, which would be another indication that we're moving from an oxic to an anoxic condition. So reality didn't meet our expectations, but it's still quite interesting. And so I wanted to look further at these basal sediments and into the upper oceanic crust to ask, how does this sediment basement microbial community change along this transect? So here's just some representative pictures of what these kind of samples look like. This is a deep sediment sample, and you can already see little chunks of kind of crustal material embedded within this material. And at the Center for Geomicrobiology, I've been working with another postdoc, Mark Lever. He's done an extraordinary amount of work to develop extraction techniques to get enough DNA out of these samples for us to work with. And so what we've been able to do so far is extract a number of deep samples from these holes and look, for instance, at the relative abundance of bacteria versus archaea based on how abundant a taxonomic gene that we're looking at is in these DNA extracts. And so, for instance, what you're looking at here is the abundance of that particular gene per gram of sediment for the bacteria and for the archaea. And this is a ratio of those amounts compared to one another. So what we can see from looking at this data so far is that in these basal systems, it seems that the bacteria are more abundant than the archaea. there's still kind of an ongoing debate within the deep biosphere community about what's more abundant in the deep biosphere, bacteria or archaea. And so this will play a part of that, I suppose. And in some of these systems, it does seem that there's a stimulation of abundance towards the basement interface. So for instance, going from about 10 to the 5th copies per gram to 10 to the 7th for the bacteria. But in other cases, that doesn't seem to be very clear cut. So we need to look at this more. Another thing that we've been doing with this DNA extract is to try and sequence all of the DNA genes, or the 16S genes in the DNA extract, to ask who's there and how abundant are those different groups. And so what we can see, for instance, just from looking at one sample from here close to the outcrop edge, is that when we look at all the bacterial sequences that we recover, the majority of them group within what's called the Chloroflexi phylum of bacteria. But you also have representation from many other different bacterial phyla. This result is not totally shocking. In other deep biosphere systems, Chloroflexi seem to be very abundant, especially in systems that seem to be getting into sulfate-reducing conditions. One of the big questions, though, is what do these organisms actually do? They're not closely related to cultivated organisms, and so really figuring out the function of this group of, or the species in this group is not very straightforward and requires more work. I want to point out that the funding for the sequencing effort that's underway, actually there's more sequencing happening like as I speak. I was hoping I'd have it for this talk, but that's how it is. Is coming from the Deep Carbon Observatory Census of Deep Life, which is funded by the Sloan Foundation. So I definitely want to thank them for sequencing our samples. So that was kind of an overview of what we are working on right now to understand life at the sediment-basement interface in a system that's relatively constrained in terms of we think we know where water goes into the seafloor and where water comes out. But obviously we're learning that it's more complicated than we thought it was. It's going to require a little bit more work. We have another project that we've been working on in the Atlantic Ocean. We recently went on an expedition to the western flank of the Mid-Atlantic Ridge. This is a site that we refer to as North Pond because it's a little pond of sediment within kind of this mountainous ocean crust area. And we need some sediment to do our coring work, so this is a great place to work. And the crust in this area is about 8 million years old, so it's still relatively young, and in a system where we think we'll find some microbial activity within ocean crust. So we recently went out there and drilled several holes and also installed several observatories around this ponded area. And just to give you a general impression, we think that water moves roughly in this direction, or north, south to north, maybe southwest to northeast. We're still trying to figure that out. It's not very easy, but we cored a number of places around the system and collected both sediments and basement. As a cross-section through this North Pond area, so if you imagine cutting through like this and looking at a cross-section, we think that water moves through this oceanic crust relatively rapidly in the upper oceanic crust, and then the fluids that are moving through are relatively low temperature and probably oxic. I'll talk a little bit more about that in a second. And so the primary motivation for this expedition was to install a number of observatories along this presumed flow path to study microbial activity in the system. Now, why do we think the water moving through the system is oxic and cold? Well, several of our collaborators have been studying the system for a number of years now. And this is a profile of temperature with depth, both through the sediment is kind of about 95 meters thick, and then you get into this basement section. And work that Keir Becker and others have done shows that you have this relatively stable temperature profile in the upper few 300 meters of basement, and then gradually the temperature begins to increase. And looking at this type of data, there's also an indication that fluids are moving through at certain areas within oceanic crust. And so I kind of consider this upper oceanic crust as kind of a well-mixed environment where you have cool fluids moving through. And one of our questions is, what is the nature of life in the system and how is it interacting with the sediments overlying? I want to also mention that this is a site that's way out in the middle of the Atlantic Ocean. It's very far away from land, and so there's very little primary production in the overlying water. And so there's very little carbon sinking down into the system, so it's relatively starved. compared to the Juan de Fuca environment I talked about before. So during the expedition, we cored into basement at a number of sites and collected some really beautiful rocks that I'm very eager for us to begin looking at. We had a nice glassy basalts, very altered basalts, some more serpentinized systems, and even some kind of hardened sediment. So this is an ongoing area of study. I won't bring that up very much. What I want to talk to you about is the work that we are doing in the sediments to try and get an idea of what's happening not only in the sediment, but also in the basement below. So one of the things that I did on this cruise was measure oxygen profiles in cores of sediment that were collected around these different locations. So what you're looking at here is oxygen concentrations on the x-axis and depth of sediment on the y-axis. And what you can see here is that you have oxygen consumption with depth in the sediment. That's not very surprising. We've seen that all over the world. Usually oxygen is consumed very rapidly in sediments and is gone within the first few millimeters to centimeters. Here it's penetrating much, much deeper, and this is because the system is so carbon starved. So you have microbial activity, but it's not very rapid. But one of the interesting things for me is that you see this increase of oxygen with depth as you approach basement. So this indicates that the fluids moving through the sediment, or moving through the basement underneath these sediments, is oxic, and is oxygen diffuses out of the basement into the sediment. And one of the things we can do with this kind of information is actually try to say, okay, if we know this gradient of oxygen with depth in sediment, can we make some predictions about what the oxygen concentration is in the basement underneath the areas where we cored? And because you have these nice linear profiles in many cases, you can make kind of a first order guess of what the oxygen concentration would be underneath the sediment column here, what it would be here, what it would be here. Then we can use this kind of information to say, okay, well, if we think that water moves generally in this direction, and we know the oxygen concentration here, or we think we know what the oxygen concentration is here, can we make a prediction about what the oxygen concentration would be along that flow path if we assume that oxygen is only being removed either by diffusion or advection or reaction? And so I developed a relatively simple model. This is actually based on work that Sam Holm and Jeff Wheat and others have developed for the Juan de Fuca system, to say, okay, if we have water moving along this way and we know what the oxygen flux is out of the basement based on these profiles, what would the oxygen concentration be out here? So it's a relatively simple model that we still need to ground truth a little bit more. But just to give you a taste of what this kind of modeling will do, what I'm showing you here is what the expected oxygen concentration would be at this site based on what we think the oxygen concentration was here. So this red line is what the oxygen concentration we think it is, and this blue line represents what the model calculated the oxygen concentration would be if the only thing removing oxygen was diffusion and advection. And so what you're looking at here, is the oxygen concentration versus the kind of flux of water through this system. And this is really the unknown in this model, is how fast and how much volume of water is moving from this from here to there. So we don't really know what this is. But we can kind of constrain, okay, well, if the flow rates were really low, then we could say that the oxygen concentration here could be explained if the flow rate was relatively very low. This flow rate is much, much lower, or not much, much lower, but lower than the rate, for instance, that's predicted for the Juan de Fuca system. And we think that the flux, the movement of water through the North Pond system is actually much more rapid than at the Juan de Fuca system. So probably flux rates here or even higher are more realistic. If that's the case, then our model would suggest that the oxygen concentration would be much higher than what we think it is. So the only way that you can bring these two into harmony again is to have some element of reaction of oxygen within basement that's removing oxygen as the water is moving along. So this is then a plot of what that rate of oxygen consumption would be per cubic centimeter of rock per day, again based on the estimates of the flow rate. So what this suggests, if this model, which is very arm-waving at this point, this is kind of one of the first indications of the oxygen consumption rate within basement. We still need to work on this model a little more. I'm just now getting other chemical data from this expedition to try and constrain what this value is a little bit better, and so that we can constrain our model a little bit better. But I think this is a first step forward, at least in trying to identify oxygen consumption within basement in these oxic systems. And the reason I think this is important is because globally, probably the majority of the ridge flank systems are these cooler systems where cooler fluids, maybe oxic fluids, are moving through. So we need to understand how quickly is oxygen consumed in these systems. Is it biological consumption or abiotic consumption? So this is a first step in that direction. I want to also give you a small taste of some other work that was done by another scientist on this expedition, Orcutt, who sailed with us, and he was applying a relatively new technology called deep UV to try and identify if we could see microbes on the rocks that we collected. So this is a figure. He gave me some permission to show. What you're looking at here is an overlay of the fluorescence of both mineral fluorescence and fluorescence thought to come from cellular material. The purple-y stuff is the mineral fluorescence. The green color represents what's more microbial fluorescence. And what I want to just give you a taste of is that from this information we can say, okay, on these different materials that we recovered, this is just a small chip, it does seem to be that there's hotspots of life. Another corroboration that there's probably microbial activity within these crustal samples. Stay tuned for more details. So coming back to this idea of, where is life in the deep biosphere, I want to again reemphasize that I really think we need to include the calculations of life in oceanic crust, and the recent data we're getting from the Juan de Fuca and the North Pond expeditions are really going to be valuable in terms of trying to give some ballpark numbers of how much life is in deep oceanic crust, and it may be very significant compared to microbial life in deep sediments and kind of change what we think about how abundant life is in the deep biosphere. So stay tuned. And with that, I have no idea how I'm doing on time, but I'm done. I want to thank people involved in this work. Katrina Edwards, obviously, I started a lot of this work while I was in her group as a postdoc, and I've continued it as a postdoc in Denmark. Thanking folks coming from her lab, Amanda, a graduate student, and Brandy and Kara, who are former members of the Edwards lab. I want to thank Jeff Wheat, Andy Fisher, Sam Holm, Wolfgang Bach, and Kier Becker for a lot of the work that we've done together on the Juan de Fuca and North Pond expeditions. Everett Salas for the work with the deep UV system, and Bill Jorgensen, my current boss, and Mark Lever, another postdoc in the Center for Geomicrobiology, for their help. And with that, I'd love to take questions. I'm not sure how we're going to do this. I think the idea was that we'll take questions from the room, maybe one or two, and allow people on the internet to type in your questions so that we can see them. And then Matt will start to relay them.
Yeah. And for those of you on the internet, if you could just enter your questions in the Q&A pod thing in the lower right, that'd be great.
Great. So yeah, first question.
How homogeneous is the rock that you're drilling? Because that would have implications both for the hydrologic flow and for the substrates for the microbes.
Yeah, so it's not homogeneous. We can see so for instance at the North Pond site, as we drilled deeper into the crust, we would go through different flow units. So you would have areas where you would have kind of more, not brittle, but glacier basalts, and then you would get into situations where the basalts were more massive. And so between those different systems, in the glacier stuff, it's usually generally much more fractured. You can see lots of veins in the rocks and also evidence that there's separation of the rocks. And then you get into more massive sections where you may or may not have fractures. So the way that fluid moves through those systems is going to be very different. And the chemical reactions, the magnitude of leaching of different minerals out of the rocks will be much different. But that aside, for instance, at North Pond, it seems that even though you have these different flow units, it still seems that that upper kind of few hundred meters is still somewhat connected hydrologically, and which for the model is important because we're kind of assuming that you kind of have a bolus of water that's moving through the ocean. Of course, it's probably moving in a much more diverse way, but we try to consider that as, you know, oxygen concentration in the upper basement in this system. How deep that goes is, you know, still something we have to learn. Another question from the room? Or are there any questions online yet? Matt or Rosa?
Yeah. So, let's see.
Oh, there may be, but we can't see them yet.
I have a question, Beth. I think you were in on the development of the flocks.
Mhm.
Can you talk about that a little bit? Where did that, what inspired you, or who got the inspiration for the Osmo chamber and the developments of that particular instrumentation?
Yeah, great question. So the flocks, which were those colonization experiments that I showed at kind of at the beginning of the talk, came together from a variety of sources. So for instance, colleagues that work in terrestrial systems, like deep mines, for instance, had come up with similar flow-through systems for addressing the kind of questions they had in the terrestrial systems. And then at the same time, colleagues like Jeff Wheat and Hans Janisch at Mbari were working on these systems which develop fluid samplers or based on osmotic pumping systems, so they don't require any power. And when I was a postdoc with Katrina, then we kind of merged those ideas to create a system that we could put in the oceanic subsurface and connect them to these osmo pumps so that fluid could continuously be pulled through these colonization experiments without requiring a bunch of batteries and mechanical pumps, etc. And then it's continued to evolve since, you know, we've gone through many beta versions of these colonization experiments to try and find the best way to do this. Okay, still don't see any questions here. Nope. People may be asking them, but I can't see them. Any other questions from the room in the meantime? Yeah, Andrew.
I'm curious why I've consistently seen so much mention of the Whitman figure of 1/3, always followed by, and this is just sediment. And I find it remarkable that there isn't some more follow-up since then to, I mean, I'm familiar with one paper that's commonly said in the same breath, but are there any more?
Yeah, so that's a great question for those who may not have heard it. Why do we keep going back to this Whitman paper, and are there other estimates of how abundant the deep biosphere is? So the Whitman and all is kind of the high end of the estimations. There were some other estimations by a group out of, where are they? Somewhere in the UK. The city escapes me now, but John Parks' group have also made similar kind of calculations, and their original calculations were maybe it's like 1/10 of biomass carbon was in cells. More recent work that's been done by folks with Steve Daunt's group and the folks studying the South Pacific Gyre are really kind of ratcheting that number down, because the original calculations were based on sediments that have been cored relatively close to land, very carbon-rich sediments where more cells can be found. But as you go out into these gyre systems, these really starved for carbon systems, the cell counts in the sediments are much, much lower. And so if you adjust your model to say, okay, well, not all sediments are equal, they're not all full of cells like these sediments close to land, but as you get farther away, they're much more, are much less abundant, then that number is probably lower. So I've seen estimates like maybe it's 1/5 of biomass carbon is in these deep sediments globally. So there are people working on it, but you know, we're kind of just making pinpricks in the seafloor and counting the cells and then trying to extrapolate for the whole sediment system. So, you know, we may be off by orders of magnitude. But yeah, but there was, I think the recent paper by Don et al. 2009 kind of addresses some of these questions.
Thank you.
Hey, questions? All right, hold your thoughts here. What do we got? So there's a question here from Amy Smith. Can you speculate on the source of ammonium in hole 1301A during the last year of incubation? Great question, Amy. She's referring to one of the quarks on the Juan de Fuca system, where we did this 4-year colonization experiment. And when the borehole rebounded, it seemed that there was an uptick in ammonium in this hole. And I would say that this is, there are kind of different camps on where that ammonium comes from. One idea is that maybe it's coming from sediment that may have gone into the hole or into the formation while we were drilling, and that as that sediment got heated up, it released ammonium, and that it's just an artifact of the drilling and observatory process, and that maybe the ammonium is not really that abundant in the formation fluids. The other end of the spectrum is that there is ammonium in the formation fluids, and trying to figure out its source would be another topic. So, maybe it's coming from organic nitrogen in the fluids that are circulating through basement and then being converted into ammonium. I think we need things like stable isotope work using 15N stable isotopes to really kind of nail that down. So that's my speculation. We have another question here from William Horsey. Did you try to analyze RNA from any samples to get a better idea of what microbes are active? Great question. So for those of you who may not think about what RNA is, in living cells, you have DNA, which is kind of the backbone of genetic information. And then as cells are growing, they're expressing certain genes for certain functions. And that kind of expression is part of RNA. And so some investigators look at both the DNA pool to see what is the potential for what organisms do and how abundant they are. And then you can also look at the RNA to say, okay, well, what's actually active? In the work that I've done on the Wanda Fuca system with these colonization experiments, I didn't look at RNA because we had very little material to work with. So even in doing the DNA work, for instance, if doing a PCR reaction, I was cutting out bands that I couldn't see. I knew they were there and worked on them. So we had very little material to work with, and working on RNA just wasn't an option. Nowadays, we're putting down colonization experiments that are much larger in volume, and so there is an interest in looking at RNA in these systems. So stay tuned. Can
you hit the webcam button on the top of the screen next to the audio?
This one?
Yeah.
Okay. In some of the...
And then start sharing, sorry.
What?
Start sharing on the video itself. Yeah,
Start sharing.
Thanks.
Something magical happened. Okay. Oh, now I can't see the questions.
Oh, I'll move it.
Okay. To come back to your question, though, about RNA, in some of the more recent expeditions, that is something we're trying to do. We, the group of scientists who collected those samples. When we collected sediments and when we collected rocks, there's kind of a group effort to extract DNA and RNA from those samples, but that's underway at the moment. Okay, coming back to questions. Where are we? All right, Ben Harrison has a question that just disappeared. Where'd it go? Am I confident that the dominant chloroflexi at the sediment basement interface are active in light of low activities inferred from geochemistry? Oh, interesting question. Wow, okay. So in the samples that where we did this sequencing effort from, there are very small gradients in, for instance, sulfate. And so if there's, for instance, sulfate reduction going on, it's probably at such a low rate that it's not really impacting the chemistry of those deep sediments. Something I'm discussing for a while. But there still may be enough energy to fuel life in the system. And so finding chloroflexi may be both looking at a record of things that were active but are not doing anything now and things that are active. We just right now have a first pass doing the sequencing effort. This is what's most abundant. And now we need to dig down into that more and say, okay, are they active? Can we get RNA out of those samples, etc. So I'm confident that we'll find an answer, but I don't have a confident answer for you yet. Kostas has a question about eukaryotic life in these systems. Another great question. So I totally ignored that in my talk. But I think this is an active area of investigation to try and understand if there's microscopic eukarya in these systems. In the terrestrial deep biosphere, there's been recent work in some of the gold mines in South Africa to collect a bunch of the fluids that are moving through these systems, and they found some really interesting evidence for microscopic eukarya, or I think nematodes, in that case that seem to be really unique and have evolved to that system and eat the bacteria in that system. They're not just coming down through groundwater, but are really part of that deep terrestrial system. So it's possible that we might also find scenarios like that in mid-ocean ridge, and hopefully folks who are better at that kind of analysis than I am will look at that. So at this point, we don't really know what the eukaryotic composition is in the oceanic crust. There's a number of investigators looking in deep sediments and finding that, at least using primer combinations that have been developed, that it seems that fungi are very abundant or are the most abundant of the microscopic eukarya, and they seem to be very different than fungal groups found elsewhere. But we're really just scratching the surface of that topic. All right, one other question we have. Julie Meyer asks, how did I stain the spiral bugs? And if I can't get DNA to amplify for PCR, are you able to use DEBI or fish stain? Interesting question. So in the Wanda Fuca samples where we saw these twisted stalks, that was actually a head scratcher for a minute of why did I see the stalks when I applied these different DNA stains, because there should be no DNA in those stalks. And our interpretation is that there was probably a little bit of grease in these samples that it had attached to the outside of the stalks during the collection of these samples, and that the DNA stain that I applied was interacting with that material, or maybe just interacting with the stalk itself in something that we can't replicate with cultures. And so the DNA, or the stain was just binding to the stalks so that we could see them, even though there was not an actual cell attached to those stalks. So we weren't expecting that, but it worked. And we could see them also if we looked at SEM, for instance, another way to look at these samples. And then, we could use, the DNA stain did attach to cells on those stalks. Can't really pull up the top now, but if you remember, I showed next to the picture of the twisted stalk, there were also some little clumps of cells. So the DNA stain does work. You can visualize cells in these samples. But there's just not a lot of DNA in this material, mainly because I was working with chips that are about that big. I just didn't have enough material to work with. I did not try fish because I didn't have enough material to try that out with. It looks like that's most of the questions I got here. So are there other questions from the room, or are we done? Yeah, well, thanks. It's been a great honor. Hopefully I looked at you in the camera enough. It's a little bit weird. And thanks for sticking through with the technical difficulties. Hope you have fun.